I'm mandated to begin on time. I know persons will come in, but I would like to welcome you to this second session of the Faculty of Science and Technology Conference, Fit for Purpose. I'm Mona Weber. I'm Director of the Center for Marine Sciences and Discovery Lab, and it is my pleasure to be a part of the conference, the 11th Faculty Conference, as well as to have the opportunity to have this commemorative session in recognition of the 50th anniversary of the Discovery Bay Marine Lab. Now, we're very proud of Discovery Bay. This is the year as well that university celebrates 70 years, and Discovery Bay celebrates 50 years. And so we want to highlight the impact that the lab has had over the years. The laboratory started really before 1968, but the buildings we occupy now and the 14 acres of land in Discovery Bay that we are on were donated to the university by Kaiser Bauxite Company. And so the main laboratory was built in 1970, and we've been operating since that time out of that laboratory. But the actual donation of land was in 1968. The university chancellor at that time, Princess Grace, received it on behalf of the university. And I must say it was very fitting to have had the present university chancellor celebrate with us on Sunday at the commemorative service. So we operate now, essentially, as you see, out of the lab. We've had numerous scientists visiting the lab over the years, but of course, our founder, really founders, Professor Tom Gorrow and his wife, Nora, were at that time physiologists in the physiology department here at UWI, but they were also coral reef scientists. And many of the research that are foundational for coral reef biology emanated out of Tom and Nora's research in Discovery Bay. Tom Gorrow was the first to pioneer the use of scuba gear in coral reef research. And they were the first to use radioisotopes to understand some of the physiology of coral. And so we have several presentations. I've lined them up one after the other. The first one will be given by Dr. Ian Sandeman. And Ian is one of the longest visiting researchers, and he still continues to come to Discovery Bay. He came in 1978 for the first time. And so this is your 40th anniversary coming to Discovery Bay. And so Ian received his PhD from UWI in 1999. Dunbar Steele was supervisor or co-supervisor, and he worked on light-related physiology of zooxanthellae, the algae that live in the coral tissues. And so Dr. Sandeman is going to speak on a method and results for the direct measurement of the calcification rate of corals under changing environmental conditions, especially pH. So I welcome Ian to take over.
I think most of you can recognize this diagram up here because it tells us about ocean acidification. The red line represents carbon dioxide in the air. About 30% of the carbon dioxide in the air dissolves in seawater, and that's the green line. And this blue line represents the changes in pH, or acidity of the ocean, that's taking place as a result. Now, I found on the other day that the amount of carbon dioxide dissolving in the sea at the moment, it's at the rate of about 1 million tons per hour. Think of that. That's an incredible amount of carbon dioxide that we are pouring out from burning fossil fuels and dissolving in the sea. And my interest is in trying to understand how corals are going to cope with that. And I feel that lab work will supplement all the things they're finding in the field, and is very important to understand the physiology of what's happening. Well, when carbon dioxide dissolves in the sea, carbon dioxide, gaseous carbon dioxide dissolves, becomes carbonic acid, and eventually converts to carbonate, bicarbonate, and carbonate, and in the process producing hydrogen ions or protons, which of course make the water acid. Now, the mixture of what you get in the sea depends upon right now the sea has a pH of about 8, and as you can see, it's made up mainly of bicarbonate, about over 90%, quite a lot of carbon dioxide, quite a lot of carbonate, and a very small amount of that red line, carbon dioxide. Now, one of the problems is what do these changes do to corals? Before I get to my results, I'd just like to let you know what the coral I'm using is an agaricea that grows in shallow water. It forms little plates. The skeleton is very nice and strong, and it's relatively simple. You can see the holes in which the polyps live, and the tentacles come out of those, not very big ones, incidentally. And it's easy to snip off on the side little bits of the coral, about 2 square centimeters is what I use for experiments, with a pair of snips, and it doesn't harm the coral, which remains in place. The corals are very simple. They just have 2 layers of cells. This is a section showing the skeleton is the brown part there, and 2 layers of cells, a large space which is continuous with the sea. It goes out through the mouth. It's the coelenteron, or gut if you like, and this is the layer that contains the zooxanthellae, these ones here, and this very thin layer against the skeleton is what forms the skeleton. The equipment I use is basically a underwater portion microbalance, which consists of a chamber with a wire, this one here. Sorry about my shaky hand, I hope you can see what I'm pointing at, which is of tungsten, kept taut by 2 little rare earth magnets, a beam with the coral suspended at one end, and at the other end a mirror. And I'll talk about that in a moment. But there's also devices for keeping the temperature constant, and I can measure the pH and get into the chamber. That's what it actually looks like, it's a photograph, and this here is a laser pointer which shines in through a wall there, glass wall, onto the mirror. And when the coral puts on a little weight, when it's calcifying, it's going to tip the beam, that moves the mirror, and the laser pointer shines a beam of light onto the mirror and then to a scale on the wall a couple of meters away, so the slightest movement will be recorded or can be seen, and in this way I can actually measure about 10 micrograms increase in weight at a time, which is quite good. Now, this is an actual experiment which shows you the help.
Sorry.
Okay, this is an actual experiment. You can see that the accumulation of weight is shown on that side. The increase in weight with time. And after an hour or so, what you've done, you've got a straight line there that you can use to get at the amount of weight change. Change the pH, and you do that by just adding a little bit of seawater with lots of carbon dioxide dissolved in it. pH goes down, and then let it settle, and you get another set of readings, and it's quite a change. And you can see in this one that it's gone from 1.017 milligrams an hour to half a milligram per hour. So there's quite a decrease with a pH drop from about 7.8 down to 7.7. So in this way you can see how the calcification rate changes if you change the pH. If you can look at the left-hand side first, what I've done here is use 5 different corals, each one with either 3 or 2 readings at different pH. If you run a regression line through all 12 points, that's the dark line, you can see that the calcification rate measured on that side is actually decreasing as the pH decreases. And there's a decrease in calcification rate of about 10% for each 0.1 change in pH. And if you look at that line too, it tells you that calcification is probably going to stop round about at a pH of about 7.5. Now I'm interested as well in what happens when you've got the same data against the hydrogen concentration, or in this case 1 over the hydrogen concentration, because you get much straight lines again. And if you take this dotted line, which is the mean of the slopes of the 5 experimental animals, it intercepts again at much the same point, pH of about 7.5. So this is bad news. If you put a coral into a different pH, you watch it slowing right down, and it decreases at quite a rate. If you think of what's happening in the world with that map, with the chart I showed you earlier, the pH is changing at a rate of about 0.1 unit every 40 years. So this means that if the amount of carbon dioxide is in the sea, then there's a possibility that corals are going to stop doing this in about 200 years' time, which is not good. But my personal expectation is that they may stop before that because of other problems like bleaching and pollution. To understand it a little bit further, there are 2 main theories about what's happening. The traditional one is that as the carbonate goes down in the sea, as the pH drops in that direction, this prevents or inhibits calcification. The other theory, which is proton flux, says that as the hydrogen increases, this line here, as the hydrogen increases, this is what's interfering with calcification. Now, the traditional means of what the traditional view of what is happening is that the various things needed for calcification are getting to the skeleton surface by a paracellular route. That means that it's not going through tissues, but is going round the tissues by a sort of route between cells. And where this happens, as the carbonate gets less, so the carbonate is going to decline. Now, there have been a set of discoveries recently that suggest that coral is able to control the pH at the surface at which it's being deposited. Now, let's just see what happens under the other theories. This is where the transcellular theory, where calcium is taken into the cellular tissue, and is pumped into the space where calcification can then take place. What I'm suggesting is happening is that carbon dioxide is the main producer, or the main source of dissolved inorganic carbon, and that in the process of reacting with the calcium, it produces two hydrogen atoms. Now these are used to pump in the calcium, and then what happens to them? Well, my view is that because calcium is coming into the cell, to maintain electroneutrality, hydrogen has to be pumped out. So I'm adding a little indication there that is what's happening. Now, think what I said a moment ago about what's happening here. If this is the route by which what is needed comes in, then quite obviously the regulation by corals of pH at the surface is going to take place. But if calcium is coming in this route, then that is not going to help the corals at all. So that's another sort of bit of bad news, if that's so. What I'm suggesting is that the regulation that's taking place is actually the hydrogen being pumped out as calcium is taken into the coral. And I have evidence for that from some experiments in which nothing happened for the first 4 hours, and then calcification takes place. But at the same time, the TA, which is an indicator of where ions are moving around, the total alkalinity, and the pH is changing. Well, why would it change? Well, it's because calcium is being taken in and hydrogen is being pumped out. That sort of confirms, gives one confirmation, and I have others. So these are my conclusions.
Thank you very much, Ian. Time for one question. We're a little over time, but quick question.
With the doom and gloom that we hear all the time, is there an opportunity for reversal? So all the talk that we... No, I know, but with all the talk about changing the emissions and so on, is that actually realistic?
This sounds like an end of session question. Yes, thank you. And our next speaker will speak about monitoring. But I just wanted to say very quickly that Discovery Bay has a long history of monitoring and unfortunately indicating the doom and gloom. Jeremy Woodley, who is here, was one of those persons who established what we refer to as LTS-1, long-term monitoring stations. And for several years, Discovery Bay has been monitoring coral cover near its lab and elsewhere, and indicating the various pressures that have been causing declines in coral cover from 70% to as little as 5%. The impacts of hurricanes literally smashing the reef has been well documented by Discovery Bay, but very noteworthy is the CARICOMP study, again, which Dr. Jeremy Woodley helped to initiate, was a study involving 27 marine laboratories, all using the same methodology to study corals across the wider Caribbean. And this is tremendous, because it meant the data were immediately comparable. They were using the same techniques, the same parameters. And this has been recently used in a seminal publication, Status and Trends in Caribbean Coral Reefs, and it's a publication in Science. Marcia and others have been involved. And so our monitoring, we think, has benefited significantly from persons like our next speaker, Bernadette, and her husband, Frederick, who is not here yet, has been studying the CARICOMP sites, have been monitoring the reefs, and they've also been developing cutting-edge techniques for accurately showing changes in our reefs. So I welcome Bernadette, and I will have to...
Good afternoon. Can everybody hear me? No? Oh, am I going to have to shout? How's that? Better? Okay. I'll try to stick close to it. Okay, how's that? Okay, I'm really going to have to speak into the mic. So good afternoon, everybody. As Dr. Weber said, I will be talking about photogrammetry and 3D modeling. But to start with, when we talk about remote sensing, just a little background, when we talk about remote sensing, we often think about products that are generated either by satellites or by survey planes flying over coastal areas, or LiDAR technology and sonar technology, which are used for mapping the ocean floor. So these products are often too expensive or are not accessible or are not provided at scales which are reasonable for projects that are done at a local scale. But with recent advances in photogrammetry and 3D modeling, remote sensing of small-scale reef studies is becoming more accessible. So when I talk about advancements, I'm talking about the increased capacity for storage and increased power, computing power of our personal computers. This now makes it possible for us to process huge amounts of data that are required to generate 3D models. So in addition to that, we also now have not exactly cost cheap, but we have affordable software solutions that allow us to generate millimeter precision 3D models. So equipped with that, we can now address some new aspects. And this is today what I'd like to talk about are precisely two examples of how 3D modeling is used by the researchers at the Discovery Bay Marine Lab, first of all, to track the growth of coral fragments in the nurseries. And the second one is, as Dr. Weber mentioned, the 3D mapping of the CARICOMP transects. So before going on, what is photogrammetry? It's certainly not a new concept. It is a discipline whereby we can extract information, we can extract measurements from photographs. So in other words, what we're able to do with photogrammetry is extract 3D information from 2D photos. And the output of this is very important because it's spatially explicit. So you get georeferenced maps, you have orthomosaics, you have digital elevation models, and 3D models of some real-world object. So in the context, the first example is in the context of the IDB Coral Restoration Project. And if you have any questions, you can refer them to Dr. Palmer afterwards. So in this study, they used photogrammetry. They used photogrammetry in addition to the traditional in situ measurements in order to track changes in the size and the volume of the coral fragments that were placed onto plugs that were subsequently placed onto nursery trees on the fore-reef of the Discovery Bay. So they, over the course of 21 months, they measured and they took multi-angle photos, and this allowed them to construct models of individual fragments in order to track how the growth and the tissue expansion changed over this period of time. So how they did this, they essentially, they went out and they would take one of these plugs and they would place it into a holder. And the reason this is important is because the consistency is important. By placing a plug into that contraption, the diver was able to go around and essentially get photos of the plug from multiple angles, from top and from the side. And this generated multiple photos. Okay, in this case, approximately 60 overlapping photos which are needed to generate a 3D model. These photos were then processed in Agisoft Photoscan, one of the software applications I referenced before. And this is an intermediate step where you can see the position of the photo. So this is where the camera was. Oops. So you can see the side and the oblique photos that were taken. And this is how many photos it really takes to generate a 3D model of a tiny little plug. So from this step, we were able to generate a 3D model. So on the right-hand side, you see the 3D model, which is a near replica of the photo. And, excuse me, the near replica of the photo that, but from all sides, which is something that you would not be able to derive just from the photo. So these models can be used to extract information. So in the bottom, we have an Acropora palmata fragment, which was modeled at month 9 and subsequently at month 15. And I don't know if you can see the numbers at the bottom here, but the program, Agisoft, actually calculates immediately the surface area as well as the volume of the plug. So this makes it very easy to calculate the change in area and change in volume. So for this particular example, the relative increase in a 4-month period, the relative increase in area was 3.2% and in volume approximately 1%. So in comparing planar measurements and 3D measurements, it's not too surprising that the 3D measurements will yield higher numbers. And this difference can be attributed to the fact that coral growth is irregular. You have lateral growth. And in particularly given the fact that you have growth along the side of the plugs. This growth, this tissue extension along the sides can be picked up in 3D models, but it's not something that can readily be detected with planar measurements. So in terms of pros and cons, there are always pros and cons. Yes, it is true. With 3D modeling, you have to generate a lot of photos. There's no doubt about it. And you have to have the computing power, and for those of us who've been working on this model, a lot of time to actually generate the models. However, the capacity to detect the minor growth and minor changes in the microrugosity makes this an incredibly powerful tool for small-scale growth studies. So in the second example, we did a 3D survey of the permanent transects that were set up at the CARICOM site on the West Fore Reef in about 6 to 10 meters of water. And this is what Dr. Weber was talking about. So these transects have been monitored, were established by Dr. Woodley, and they have been monitored since 1993, so there's a lot of legacy data. So this provided a perfect site for us to go and experiment with 3D modeling. So when it comes to building a 3D model, there is a very intensive process, and well, as you saw from the previous example, there is the data acquisition component and there is the post-processing. So for the 3D models of the transects, we set out to survey each one of the transects. And the first thing that we had to do was actually geo-reference the starting point and the ending point of these 10-meter transects. And in order to do that, we placed visible markers at the beginning and at the end of the transect. And then we used a tethered GPS to get the precise to get the precise GPS location for the start and end point. In addition to that, we placed a reference scale, a meter-long reference scale in the middle of the transect, and along with the GCPs, the ground control points, and the yardstick, or the meter stick rather, we were able to establish a scale for the entire model. The images were acquired using a video camera, a very sensitive video camera because you cannot use a flash, and a 4K camera, so the images that we extracted were very high resolution. So the post-processing has a number of steps. There's image extraction, there's mesh building, and then there is the output of various products. So I'll take you through each one of the steps. So as you can see here, the camera, so the video was taken at nadir, so right above the transect, and then at 45-degree angles on either side of the transect. The videos were then used, were processed in another software application called Pix4D. And we used Pix4D to extract approximately 300 photos, 300 overlapping photos, ensuring that there was at least a 60% overlap, because this is what you need to build the mesh. So once the first, actually I should go back, the first step, as I was saying, the overlap is very important. The first step is actually to aggregate these overlapping, or these points that appear in multiple photos, and you construct, oh wow. Okay, I'll try to speak really fast. You construct essentially a framework upon which a mesh can be built. So this is the mesh. And this is the final product. So this is what the final model looks like. This is T4 for anybody who dove on the sites. There's no audio. There's no audio to this. It's purely a visual. No audio whatsoever, and it's done. Anyway, very quickly, the surface that you saw before, the mesh that you saw before, along with this, these are products that can be exported into GIS applications and into CAD applications for further analysis. So the orthomosaic is the primary output. This is geometrically correct. So you can go in and you can measure individual, you can actually extract photos from it. And this is an example of the resolution that you can get from the orthomosaic. The next product, and this one is kind of neat because it allows, this is a digital surface model, which is a numeric representation of the rugosity. And this is what, this layer was of interest because we were able to emulate the link chain methodology that is used in CARICOM to calculate rugosity. And we did this by bringing in a dense cloud into another application called Cloud Compare. And from that, we were able to generate a contour line across the middle of the transect, which then allowed us to extract a virtual chain link, which allowed us to calculate the rugosity of that particular transect. Orthomosaics can be compared from one year to the next. Here we have 2016, the surveys that was done in 2016, and then the survey that was done during the bleaching time. And what's cool about this is that you can actually virtually tag photos and follow the fate of a single photo between the years without having to go out there and place a tag or handle the corals. And for anybody who's ever had to tag corals, you know what it's like to go back and try to find one. So very quickly, this is a virtual tagging and very powerful. We're just in the process of analyzing these samples from last year. So the 3D modeling, photogrammetry and 3D modeling has a lot of implications, and it can be used as a tool to enhance various aspects of coral reef research. The list is here, so I'm not going to belabor that. I would just like to thank Peter, who's been working on the 3D models with me, Dr. Palmer, of course, Dr. Weber for making this happen, and all the staff at DBML who are always there to support us in the field. Thank you.
We're going to move on through the talks and then perhaps save time at the end for a few questions. Discovery Bay continues to monitor, but very importantly, as sea surface temperatures increase, we've been noticing bleaching. There is now a bleach watch. Every year, NOAA sends out information as to how much bleaching we're likely to have because of the temperatures of the seawater as they continue to rise. And so our next presenter has been working on coral reefs for a long time. It belies her youthful appearance. And she has recently been doing work on coral bleaching. And so I'm going to call Marcia Crary, who is a graduate student in the department, as well as our environmental data manager, to give her presentation on bleaching and increased sea surface temperatures of Jamaica's coral reefs.
Good afternoon, everyone. I'm going to try and be as efficient. My boss has told me I have to be very efficient. So you see the title there, Increased Sea Surface Temperature on a Jamaica Coral Reef during the 2015-2016 mass bleaching event. This is on the North Coast. Now, I'm going to quickly go through the importance of coral reefs because you've heard us talking about coral reefs, but we maybe have not explained how important they are. Coral reefs occupy only about 1% of the Earth's surface, but they are responsible for approximately 25% of marine life. Now, they live in optimum, the conditions that are optimal for them are temperature, water temperature between 23 and 29 degrees Celsius, salinity, seawater salinity, and upwards to 42 parts per thousand. And they're restricted to the euphotic zone at about 70 meters max. They're very important for coastal protection. They provide habitat and shelter for a lot of marine organisms. They provide for our artisanal fishers, as you can see in a previous presentation, how important the fishing industry is. They provide food and employment for vast numbers of persons. Of course, tourism, diving, and they're also a place where we can find many bioactive compounds that are used in cures for various things. The impact of global warming on coral reefs has been quite significant, and there are 4 things that impact the corals as it relates to global warming. One of them, and probably the most important, is the sea surface temperature, which provides thermal stress for the corals. Ocean acidification, as Dr. Sandeman spoke about in his presentation, where reduced pH by some means results in the reduced abilities of corals to calcify and to produce their coral skeleton. Of course, there's sea level rise that also increases the amount of sedimentation and is likely to smother corals, and changes in the patterns of storms and hurricanes and so on can do significant damage to coral reefs. In addition to those ongoing problems that they have with overfishing, land-based sources of pollution, and coastal development. So what is bleaching? We've heard about bleaching, and this is when the corals are actually under stress. And I say thermal stress is the main thing, but there are other things that can stress the corals that can cause them to bleach. But the thermal stress is the main one that we are concerned about now. During that time, they expel their symbiotic zooxanthellae. These are algae that live within the tissue of the corals, and they provide a symbiotic relationship with the exchange of food and oxygen and so on. And the tissue of the coral becomes pale or white. Now, this does not mean that the corals are dead, but they're under a condition of stress. And not all species will bleach, and not all species that bleach will actually die. But they have found that once a coral colony bleaches, it's likely the next year that it will bleach. It becomes more susceptible to coral diseases, and generally its immune system is compromised. So in this regard, the prognosis for long-term survival of corals is not very good. So mass coral bleaching is a recent phenomenon in the last, say, 40 years, and we've had global mass bleaching 1998, 2005, 2010, and again 2015, 2016. So we've had Caribbean-wide bleaching reported, and also for Jamaica. So as Prof. Webber mentioned, NOAA has a site in which it has this product that can predict when bleaching will take place. And so for 2015, they predicted that we would have bleaching in Jamaica. In fact, they predicted mass bleaching worldwide. It started in Australia on the Great Barrier Reef, and it was expected to also happen in the Caribbean. So I guess we took the opportunity to see, if it would happen here. So the objective of this study was to determine whether there was ocean warming, because this is what causes the bleaching primarily. Did the corals bleach, and to what extent did the corals recover from this bleaching? So there are a number of sites around Jamaica that was involved in this study. This Negril and Pillow Island, right next to Alligator Head, where temperature data is available. These two sites, one at Discovery Bay and one in Port Royal, was where the surveys were done to see if other areas had bleaching, and most of the I'm in the wrong direction. I'm so sorry. Most of the sort of underwater work was done here at Pillow Island. This is Alligator Head, and this is Blue Hole, so this is important. Okay, so the methodology, sea surface temperature, we got satellite-derived data from NOAA's website. It's freely available. Anybody can download it. Received Hobotem data logger data from NEPA, who had a project to monitor the sea surface temperature at a number of locations around the island. Fortunately, Pillow Island was one of those locations. To assess bleaching prevalence, we just visited a number of sites to see whether there was bleaching or not, which species bleached. At Pillow Island, we did the benthic cover assessment, where we did 10 transects, did some photoquadrats, and used CPCE to assess the percentage cover of the various benthic components. And also to assess recovery, we tagged a number of colonies and watched their progress over time. So the first set of results, the top graph shows data from NOAA from 2001 to 2016, and this is the mean temperature at their virtual station at Negril. So what you can see from that is a trend line of increasing mean sea surface temperature. So temperatures are increasing. In the second graph, we have from 2013 to 2016. I've extracted again the NOAA data and superimposed on it the data that we got from NEPA, and you can see that there is a good match between the virtual data and the in situ data. And again, even in that shorter time frame, you can see that there is also some amount of increase in temperature. I just want to point out that at 30 degrees Celsius is the bleaching threshold. This is where NOAA predicts that at that temperature, bleaching is more than likely to take place. So the survey, we have 3 locations that sort of general assessment was done. And what you can see here are the number of species that showed some level of bleaching, so either mild, moderate, or intensive. You will note that there's these 4 species that are highlighted in red have showed bleaching at all of these locations and at both these occasions. So this is 2015, 2016. They had bleaching, and most of it was moderate to high. Those that are highlighted in red, I have also looked at them in detail in terms of tagging them and watching their bleaching and recovery. With respect to the benthic substrate, so assessments were done on 4 occasions, and this is the composition of the benthic, of the benthos. Here you see, this is the coral cover. So as you can see, coral cover is less than 25%. Again, here you have sponge. The majority in green is macroalgae. This is calcareous algae. And, sorry, what is it now? Sand, yes. So you see that corals right now, they make up, and this is a good site. This is a site that has good coral cover. It makes up less than 25% of the substrate. In the bottom graph here, what I have shown is that this is taking this and expanding it out now. So of the coral cover, you see that this is the amount of that coral cover that showed bleaching. In 2015, you see that there's bleaching, and this bottom here is dead coral algae. As the coral cover improves, or as the bleaching reduces, the dead coral and algae increases. And again, there's another bleaching event in 2016. So we tagged a number of coral colonies, and Bernadette spoke about how easy it is or not to tag and follow the progress of a coral species. But what I wanted to show here was that this is November when we assess the proportion that is bleached. So this is live and healthy, this is live and bleached, and this is dead. So you can see as they recover, the proportion of bleach is reduced. There's some amount of recovery, but in this particular species, there's quite a large amount of the coral colony that has died. With Orbicella, it's a similar sort of thing, a large amount of bleaching. It has recovered over time. At the end of that period, 8% of that colony had died. With Pseudarastra, you see a lot of bleaching, a quite quick recovery, and a small amount of that colony had died. So you see, and let me just say this one, Orbicella, bleaching, and recovery. What this shows is that different species, whether they're plate species or boulder species, react differently to temperature, they bleach differently, and they also recover differently. So you can't have like one blanket statement about corals and bleaching. So we are concluding that increased surface temperature did take place. For 2015, the temperatures went as high as 30 degrees. 2016, 30.3. So Jamaica did experience wide-scale bleaching. At Pillow Island, 23% of the corals bleached in 2015, 14% in 2016. And as I said before, species exhibited variable bleaching and recovery patterns. And I think I'm done. Yeah. In my time.
Okay, thank you very much, Marcia. I have a minute or two. Any burning questions relating to bleaching and corals. Sylvia was first, and I'm allowing one, sorry.
The thing is that there is just, there was an anomaly in 2016 where we had Hurricane Matthew. And there is a thought, I mean not proven, that that might have sort of not made the bleaching or the temperature increase or the period over which the temperature was high as intense. They have a station out in the middle of the Caribbean that said it was a 3-degree decrease in the sea surface temperature at that location, way out in the Caribbean, but that might have had some impact on the reduced bleaching effect of the increased temperature.
Okay, thank you so much. Our next speaker will focus on the topic of the lionfish. If climate change was not enough, our reefs are also being severely impacted by lionfish. Dr. Dane Boodoo pioneers research in this area, and he and his team have been demonstrating how destructive this animal can be on our reefs. Came into the Caribbean through various means, but the red dots, you can quickly see how the lionfish has spread across the wider Caribbean and America. I think we first saw lionfish on the North Coast in 2008, and now they're everywhere. And so our next speaker is Denise Chin, who is a graduate student in the Department of Life Sciences, working out of Discovery Bay and supervised by Dr. Boodoo and Dr. Aiken, will speak to us about how they have worked at solving the lionfish problem.
Good afternoon. Today I'll be talking about solving the lionfish problem through research and innovation. The presentation will take the following outline, where I'm going to talk to you about lionfish, what it is, what the problem is, and how we went about reducing the populations of lionfish in Jamaica. So lionfish is an invasive alien species. What does that mean? It means it's not from Jamaica. It is actually from the Indian and Pacific Oceans, and it is thought to have been introduced into the Atlantic and Caribbean through the aquarium trade. It is from the Scorpionidae family and has very few natural predators in the Caribbean. It has venomous spines. It has 12 to 13 on the hard dorsal spines, 2 on the pelvic, and 1 on the anal fin. They reproduce very rapidly in our waters, which is they are reproducing at a higher rate than in their native area. In fact, lionfish in the introduced area can reproduce every 4 days year-round and produces up to 2 million eggs annually. They are voracious feeders. A study done by Albin and Hickson in the Bahamas showed that they actually reduce recruitment of juvenile native fish stocks by up to 79%. And so we first saw lionfish recorded in Jamaica in 2008, but officially in 2006, but official records show 2008. And from that, we had a regional project called Mitigating the Threats of Invasive Alien Species in the Insular Caribbean, a very long name. And that gave birth to the Lionfish Pilot Project, which was housed at the Discovery Bay Marine Lab. The research components, there are 4 specific research components. Today I'll be focusing on component 2, which is the prey preference. We did our study in Discovery Bay, and we focused on 3 sites. They are called Skeggy Reef Dairy Bull and Skeggy Reef Dairy Bull and Pear Tree Bottom. And we also collected information along the coastline itself. So lionfish were also collected along the coastline and also within the fish sanctuary. And we had a special permission to do that. So we collected lionfish using scuba diving and using a pole spear, as well as collecting with the traditional pot fishing. We use the Antillean Z traps, which are normally used in the artisanal fishing industry. We collected data such as total length, standard length, wet weight. We also wanted to collect information on gape height, gape width, along with other data. But most importantly, we wanted to look at what is lionfish eating. So we analyzed the gut contents of 1,498 lionfish stomachs, and we tried to identify it as much as possible to species. So we found 22 different families within our study, and about 25% of those belong to the Holocentridae, and most of us will know the squirrelfish, which is the bony fish. They made up a very large component, and then followed by the little shrimp like peppermint shrimp or your banded coral shrimp. But we wanted to make the data a little bit more easier for us to relate to. So we grouped them into finfish, which are the teleosts, crustaceans, and we did find a one-off squid in the stomach, which is represented by mollusk. PD, which is partially digested proteinaceous matter, is a composition of all three groups, but they have begun to break down, and as such, we weren't able to classify what the species was or the group it belonged to. So as you can see, 69% belong to the finfish. What does that mean? It means, therefore, that we have to protect. We are already overfishing our stocks, and the lionfish as an introduced and invasive alien species has the potential to further reduce our native fish stocks, and in particular, juvenile fish. We analyzed the data across the size class of lionfish, and again, the lionfish 100 centimeters and over, all of them had 50% of their diet composition, which were juvenile fish. And as you can see, lionfish that were greater than 400 centimeters mainly fed on finfish, and those are juvenile finfish or small adult fish such as gobies. The top 10 predators we found, the squirrelfish, the banded and peppermint shrimps, wrasses, gobies, damselfish. But of importance in the top 10, we have our parrotfish, we have the snappers, and we have the groupers. So how does this help the innovation aspect of it? How does it help to reduce lionfish population? The research gave information on what exactly it is doing. It actually showed us the different species, ecologically and commercially important species that it is consuming, and as such, we developed a public education toolkit. The toolkit included posters. It also included preserved species of actual specimens that we collected from the stomachs. And this was very important because persons related a lot to seeing the specimens itself. And as you can see here, we have the parrotfish that is represented here, the groupers, and the snappers, which I'm sure a lot of us like to consume. So again, the analyzing the gut content is very important to see if it is actually going to be destroying commercially important species or ecologically important species. And those are from our native fish stocks. Another component of our toolkit is that we wanted, because the lionfish is venomous, we needed to ensure that our stakeholders can handle and clean the lionfish. And as a part of this public education, there was a niche market that was developed. We also wanted to ensure that the persons that we train, such as fishers, they know how to identify symptoms of being stung by the lionfish and how they can effectively treat lionfish stings. So this was all a part of our public education toolkit developed by the team. Now, outreach is very important, and one of the very good things about this project is that we had a lot of public-private sector partnerships. And in this picture, we created a submarine on land. And this was a guided tour which gave an opportunity for members of the public from various backgrounds an opportunity to see what it is, what kind of marine life that we have in our waters. And then throughout the tour, it will indicate, it will introduce them to the lionfish and the problem that we're having with lionfish consuming all of our native fish stock. So it is giving them an opportunity to, one, identify with native fish stocks and see how beautiful and important or natural local biodiversity is, and this introduced species coming in to destroy our native fish stocks. So it is an opportunity for us to give, to share the knowledge that we have. Another thing that we have been doing is we have been engaging non-governmental organizations because we believe that we have to build capacity, and once the project and the research component of it has ended, we want sustainability. And so we engage persons like the Montego Bay Marine Park Trust, members of the hotels, members of fish sanctuaries, and we show them how to safely handle and prepare lionfish, as well as giving them again the opportunity to relate to the gut contents and seeing what exactly the lionfish is eating and the problem that it is creating. We did talks with the Brownstone Fire Station because first aid is very important, and a main group that we had a talk with are fisher folks on the Pedro Keys. I know Trench and myself went out there, and it was very important that we targeted this group because they are very important to the commercial fishing industry, and they assisted us with starting the removal of lionfish off the Pedro Keys. Now, what we wanted to tell everybody at the end of the day is that, hey, lionfish can also be eaten, and we show them ways in which they can prepare it. Ways like that we are familiar with escovitch, they could roast lionfish, so it's things that they could relate to. We also developed through the project a Let's Eat It to Beat It campaign, and at the end of the day, we wanted to let people know, eat a lionfish and you're saving up to 20 juvenile native fish by eating one lionfish. And we have found 20 juvenile fish in one stomach, and other countries are reporting up to 30 juvenile fish in fish stomachs. So in conclusion, we found that lionfish diet is very diverse. They're not specific to one group. They're not specific to one type of fish. And we also found that the majority of the gut contents was composed of finfish, teleosts, and then to a lesser extent crustaceans. Fish larger than over 400 centimeters were preying mainly on finfish. And we did find that communicating the science to the public works. This helped us inform the public that you can help to reduce lionfish and save our native fish stocks. And in fact, our research throughout the years have reported a reduction in lionfish in Jamaica through the research and public education. Thank you. I'm sorry.
Thank you very much. Thank you, Denise. I was trying to look as menacing as possible. Okay, our last presenter is going to speak on rehabilitation. And in light of the demise of both corals and other natural ecosystems, Discovery Bay has taken on a lot of reef rehabilitation, a lot of coral nursery work. Bernadette alluded to that in her talk. But we're going to focus, we have a poster actually that I want you to take the time to look at on coral reef rehabilitation done by Peter Gale. But our last talk will focus on mangrove rehabilitation. And Mr. Camilo Trench is a graduate student in the Department of Life Sciences, but he is also the Chief Scientific Officer at the Discovery Bay Marine Laboratory. And I'm going to ask Mr. Trench to come and speak to us about effective mangrove rehabilitation methods in Jamaica.
Okay. Good day, everyone. Yes, and thank you. Yes, Ms. Chin, as usual, lionfish hugs the attention. No problem. We caught 132 in Discovery Bay this week, so the numbers are reducing. Okay, so mangroves in Jamaica. So my talk is mangrove rehabilitation, methods of mangrove rehabilitation, and we'll get straight into it. So mangroves, just as an introduction, mangroves are flowering plants. They dominate the tropical coastlines. They have special adaptations for these harsh conditions. These are mostly harsh coastal conditions as well as saltwater regiments. They have viviparous seedlings, meaning that the seedlings are they are partially germinated even while attached to the parent. They have waterborne dispersal, so the seedlings are dispersed by water primarily, with the exception of the buttonwood mangrove, which is really a partial mangrove. It's really not a true mangrove species. So Jamaica really has 3 true mangrove species, which are the red, the black, and the white. And the buttonwood is a mangrove, a minor mangrove, or some persons regard it as a mangrove associate. They are facultative halophytes, meaning that they do not require salt water, but they have mechanisms to deal with the salt water, either by excretion of the salt from their tissues or exclusion of it from their bodies, especially the red mangrove. They have specialized roots and spongy tissue called erenchyma. And they have lenticels which allow them to breathe better in the waterlogged environment. And they have support roots and buttresses, as identified by earlier scientists. So mangroves and coastal forests in Jamaica go hand in hand with our development. Our 2 major airports were constructed in mangroves, numerous marinas. They have retented billions of gallons of flood water. You've never heard about the Black River flooding because of the mangrove and the wetland system down there. And not much tourism, so it's still relatively healthy. Ecotourism, Black River Safari earns millions of dollars and supports a lot of persons down that side. Big nutrient filter in places like Falmouth and Hellshire. And if you come from Portmore, Hellshire side, more than likely your sewage treatment is partially filtered by mangroves before it enters that bay. Windbreak for various shipping interests, whether cruise ships or bulk or transshipment ports. And of course, recreational and cultural, very important. Some persons go to crab bush, some persons go bird watching, etc. And majority of our coastal towns have mangrove origins, many of which we don't know what they looked like originally because they're all dumped. So mangroves have over 200 documented uses, and I didn't want to give you a long list, so I just put this together. That is the Kingston Harbor, and you know, the Kingston Harbor, you notice the one in the middle here, transshipment, that makes a lot of money for us, some years more than tourism. So these are all the different functions that the mangrove forest provides for us within the Kingston, Palisades and Port Royal protected area. So sand dunes here, mangroves all around on the inside, and it provides all of these services. Filtration, of course, it keeps the manufacturing industry nice and calm. Lots of energy is produced there. It has cultural uses in the mangrove. It's a heritage site, and I could go on and on and on. So you get the gist. Now, in terms of mangroves, they are certainly in threat, and most of them have been lost through reclamation projects, especially related to mariculture. So 52% of the world's mangroves have been lost through mariculture. That is stuff like shrimp farming primarily. So when you eat your shrimp over in Glorias or wherever, bear in mind you could be eating shrimp from a previous mangrove forest from China or Singapore. That is the reality. We do not have a whole lot of mariculture in Jamaica, and sadly, sadly many of our shrimp farms have closed down. That is a bittersweet thing. We're getting more imports, less mangrove destruction, but let's not debate that. So mangroves are also degraded through natural catastrophes. Hurricanes do a whole lot of damage, and tsunamis. But bear in mind, they also protect persons from tsunamis. For example, the Boxing Day tsunami in 2006, where mangroves protected hundreds of thousands of lives in the lesser developed areas. Specifically for Jamaica, Jamaica has only about 2% of its landmass as coastal forests, most of which are mangrove. We've lost over 2,000 hectares of mangroves between 1989 and 2010, and this comes from the NEPA State of the Environment report. Quick case study down in Trelawney. Trelawney, one parish alone, has lost over 160 hectares of mangrove forest in 5 years. And we can see images from one of the hotels that is being built there, Oyster Bay, and essentially a road goes through the mangrove forest. And it's funny enough, in putting this presentation together, when I used this photograph, I saw that the color here was different from the color over there. And I immediately called my colleagues at NEPA and said, did this road have culverts? And no, it didn't. So I hope they're going to remedy that now. So, of course, they already killed one section of the mangroves, and by not having culverts, the rest of it is guaranteed to die very slowly. So here we have the mangrove areas in Trelawney. So this is definitely under threat because St. Anne is already developed, St. James already more is developed, so Trelawney is what is left on the north coast. And believe you me, we have a lot of nice white sand beaches which are occupied by a mangrove forest down there. I hope they'll remain good secrets. So, in an effort to reclaim some of what we have lost, we try to do mangrove rehabilitation across the world. So, majority of these restoration attempts have failed, and this is well documented by our first world scientists, if you want to call them that, especially in Florida, Asia, etc. And persons believe that you can rehabilitate a mangrove forest by just putting plants in the ground. So they often wonder why doesn't it grow. And here we have photographs of persons putting mangroves in rows, nice plantation attempts, and most of them have actually failed, and billions of dollars have been lost by trying to plant mangrove forests. So this is widely attributed to mangrove gardening, and this is what it's termed by Robin Lewis, which is one of our foremost mangrove rehabilitation experts worldwide. He's based in Florida. So conservation of mangrove forests should be key, and it certainly should be a priority over rehabilitation. And Jamaica has made some attempts. We are signatories to the Ramsar Convention. We have Ramsar sites. We have been doing some work in that regard. But our ecological restoration is basically regaining the functions of the forest basically from any prevailing physical factors that were preventing the forest from regaining its pre-disturbance characteristics versus a rehabilitation, which is more like a partial reclamation of some of these. However, rehabilitation doesn't give you a full restoration type of, it's a little bit different. Rehabilitation is en route to restoration in other terms. So we'll get some of the pre-existing conditions, but we'll never return to what it was. That is what we are attempting, rehabilitation. Restoration will take maybe many generations. So what do we do to look at our mangrove forests? And this was primarily funded through the Environmental Foundation of Jamaica. I must big them up. We gathered background data on these mangrove sites. We basically chose these mangrove sites based on satellite images as well as information from NGO partners and government agencies. We did assessments of the vegetation, the aerial satellite images, salinity, the detailed vegetation surveys in 10-meter plots in most of these areas. We looked at species composition, number of mangrove plants to get an idea of the density of the trees, the DBH, canopy cover, etc. Very important, we also looked at the tidal heights, where we have data loggers, Hobo data loggers, very useful, and they record how much tidal influence these forests are having. And which was quite new for us and novel. We brought in topographic surveyors into the mangrove forests. So the same surveyor that would survey your land or development, we brought them into the mangrove forest to look at the level of the mangrove forest to see, is it too high, is it too low, why is it different from a healthy mangrove forest? We couldn't do that by just eyeballing it because, of course, we want it to be scientifically sound. Now, experts across the world, and we did agree based on what we found, is that hydrology, or the movement, the net movement of water across the land, is a major factor which causes mangrove mortality or prevents the mangroves from actually recovering. Now, though mangroves have some natural mortality, unnatural mortality is mostly often related to hydrological issues. We have anoxia, some of them getting too dry, desiccation. Now, entanglement, toxic exposure, these are a little bit different. And then, of course, we also have catastrophic events like breakage from hurricanes. Other issues that prevent mangrove forests from recovering, we have grazing of seedlings by things like goats. Hurricane damage, we spoke about that earlier. For example, Hurricane Hugo wiped out majority of the mangrove forests in Guadeloupe. And a fair amount of, which is fairly new, maybe the last decade or two, solid waste is a very big problem now. Breaks the mangrove seedlings, tangles them, etc. So, based on our findings, it is clear that rehabilitation of mangrove forests need to be variable and specific. It cannot be a cookie-cutter kind of thing, and not all mangrove sites will be different. So based on some of the sites we've been looking at, some in depth and then others that we've worked with other government partners or NGOs, we need to do slope corrections in some areas. Some areas need to be cut, some areas need to be filled to actually regain it to the proper mangrove slope and topography. And what I didn't put in my presentation here is that healthy mangrove forests in Jamaica have a gradient or a slope of between 0.5% up to about 4%. If it's anything different from that, it's going to be a struggling mangrove forest. So those are the benchmarks, between 0.5 and 4%. Solid waste segregation sometimes is very necessary. Solid waste comes in and it destroys the young seedlings, and we've done that on the Palisados. We'll do it on the airport runway, and we also have a site in St. Elizabeth called Malcolm's Bay, where the solid waste, we never had anything else to term it, were actually trees, large tree trunks. So we had to remove those as well. So it's ironic, we actually bring a chainsaw into the mangrove forest to cut out the dead tree stumps, and it helps. Human management, of course, human cause many of these issues. Adhering to the building permit system, charcoal, burning, community outreach, and in some cases fencing, like in the case of Portland Cottage, where NEPA undertook that project and we provided support. And recently we were down there, we saw the fences being breached by the goats. The goats were literally pushed their head through the fence. They're still trying to get at the mangroves. Just a couple of our case studies. That's Lilliput. That's a site where we started looking at it in 2012. That's a site after we scraped it down last year. And then that's a site a couple of days after it scraped down. And then a couple of months later, it's tidal. So the trees haven't started to really recover yet, but now it's getting water. So the hydrology has been restored. So we'll study that site for about another year. One thing interesting is that this is the site, and all of this area, if it wasn't rehabilitated, what would happen is the same kind of pattern that happens all around that North Coast area. Persons start to build illegal structures. And this is actually 25 acres. We did a calculation. So you can imagine 25 acres in informal settlements in the mangrove. It's quite possible and very easy. And our parish council isn't doing much to stop it, believe you me. That is the Palisados, one which is a little closer to you. We had 70% survival in 12 months. We had flowering black mangroves. We're monitoring the height, number of leaves, etc. And the faunal recruitment was certainly improving. There was nothing there when they graded it. We're having now snails, fishes, birds, and people doing fishing. And of course, sadly, the government agency which we had a contract with didn't provide the money for us to do the cleaning. And now that is what it looked like at month 12 versus month 18. And the seedlings cannot survive because they are smothered or broken or entangled by the garbage. And the survival dropped to 40% in 18 months. And 70% of this budget was actually to clean the solid waste. So that is my take on mangrove rehabilitation methods. Thank you very much for listening.
All right, ladies and gentlemen. I know your program said something about past, present, and future as a wrap-up talk. But essentially, I've been weaving through the various presentations information about Discovery Bay and about what it's been doing over the years. And if you're willing to go a few minutes into your tea break, we can take a few questions. So, yes, one at the back. Daniel. Any other questions?
Camillo, you said that over 2,000 hectares of mangroves have been lost in that short period of time. What was the total area? In other words, is that 10% or 20% or 5% of Jamaica's mangroves?
I really didn't do a calculation. Sorry about that. But I would guess somewhere between 20 to 30% of the mangroves in that period of time.
That's a really significant number if true, so I would encourage you to make that calculation.
Yes, we shall.
Mine is a community question because I happen to reside in Cardiff Hall, so I can walk across to the beach by FDR, which I have sometimes done. So people in Cardiff Hall are not aware of the lionfish hazards. There's a Cardiff Hall community, and some time ago I did try to reach the Discovery Bay Lab to get some information, to give a talk to our community. But I don't know what happened. The communication lines failed. The link in the Cardiff Hall community, I don't know, she said there was some problem getting through. So given that it's on my doorstep, and my partner, who is every time I go, she says, Lloyd, you see what's happening? They're taking away Jamaica, new Spanish hotels. So, there is concern. So your work is very valuable, but you need to communicate. Anyway, I'm here talking to you. The fact of the matter that lionfish can be eaten, I did some research. My partner and I, we spent money, and we spent a lot of money researching. So we know that. But it's no use. We can't tell them. But if we could link with you and share this information, then it would get through to the community. You want to do real community outreach. Anyway, this is our gesture.
Thank you. It's interesting because I had a few slides on what else Discovery Bay does. And a significant aspect of our work now deals with community outreach. We bring the public in, we go out to the public. In fact, Mr. Trench is our main outreach officer. And so we're very aware of the need for partnering with the community to really see effective rehabilitation and conservation. Okay, there's one last hand at the back. Dane, sorry, Dale.
She used to be in social work, so she's very, very interested.
Good afternoon. My questions related to the lionfish talk. Just want to know if consideration has been given to genetically modifying the lionfish in the same way that mosquitoes have been used as in the past year to either prevent them from laying as many eggs or to reduce something.
Well, you know, I'm very cautious about genetically modifying anything, much less an invasive species that's already detrimental. There has been some work in looking at sterilizing lionfish, you know, where you can actually have them producing only sterile offspring that will never develop and start reproducing. But that hasn't really gotten anywhere, because not much is known about the lionfish. Believe it or not, I mean, about a couple hundred papers written on it in this region. That's only in the last 10, 15 years. Nothing has been known about it from before. So there's a lot less known about it right now that you can then qualify it. Let's go GMO or let's modify something to help control it. Right now, the control mechanism, as Denise pointed out, is, you know, consumption with the aim of reduction. So eat the small ones, eat the big ones, eat every single one as much as possible. And that has worked. It has worked. It has worked. We've seen it in Jamaica. We've seen it all, and it tastes good. Thank God it tastes good. All right? Thank you.
Okay, thank you very much. But one last.
I just wanted to throw out a question to Camilla Trench for the areas that the areas that are polluted with plastics. Have you seen any virological or bacterial diseases in the mangrove plants there? Just an off-ball question.
To be honest with you, that is not my specialty. We have seen lots of things, but that would be something for the microbiologists to come out and study with us. We really don't know what we're looking at. We do have some cyanobacteria that accumulates when there is stagnant mangrove waters, but that's nothing that is really unusual for mangrove rehabilitation projects. But that isn't really in keeping with the new solid waste. That is normally just stagnation by itself, not just caused by plastic or anything. All right?
Okay, well, thank you very much for coming and staying. And we continue our celebrations in Discovery Bay on Friday. We invite you to the open day if you can make it. And we close this session. And thanks to the Faculty Conference Organizing Committee for allowing us this commemorative session. Thank you.
And just before we go, just a reminder, after the break, we're going to have parallel sessions. One on ICT and modeling and SLT2, and the other one on energy and materials in here. So have a good coffee break.